Hello everyone, we are a bioprocess engineering group in EECE 506 from WUSTL. In this YouTube video, we are going to show our crazy idea about converting waste to energy on Mars using a bacteria called Geobacter. This idea is proposed based on the vision of a successful human landing and starting space living on Mars. Before getting far, let's take a look at the difference between Mars and the Earth where we live. First, the atmosphere. Mars has a much thinner atmosphere than the Earth. The surface pressure on Mars is about 6 to 7 millibars on average, compared to 1,000 millibars on Earth's surface. Also, the composition of the atmosphere on Mars is totally different from the Earth. We've listed 3 main components on Mars and compared them with that on Earth. The Earth owns 78% of nitrogen, 21% of oxygen, and 0.04% of carbon dioxide. However, on Mars, over 95% of the air is carbon dioxide. The nitrogen is less than 3%, and there is only a trace amount of oxygen. Second, the gravity on Mars is much lower than Earth's gravity. Third, Mars is much colder than Earth. Since the Earth's average distance to the Sun is about 93 million miles, we call it 1 astronomical unit. Mars is 1.5 astronomical units away from the Sun. That results less solar radiation and lower temperature on Mars. The temperature on Mars ranging from negative 158 to 20 degrees Celsius, while the temperature on Earth ranges from negative 88 to 58 degrees Celsius. Lastly, the soil composition on Mars also has a huge difference from Earth's soil. The soil on Mars is almost entirely made up of minerals, with only 2% of water and air and no organic matter. While on Earth, half of the soil is made of air and water. It also contains 5% of organic matter and 45% of minerals. Since there's no organic matter on Mars, technically speaking, it is not real soil. The proper term for the surface material on Mars is regolith, which is a broad term for the loose material covering the surface of the planet. And soil is a type of regolith. Here's a list of the mineral composition of the Martian regolith. We notice that the ferric oxide takes a large portion, up to 18%. This makes us wondering whether we can utilize this material to do something interesting. Geobacter bursts into our eye shots with this amazing property. It can convert ferric oxide to ferrous ions and generate electricity. Given the conditions on Mars, we decided to use Geobacter to achieve the goal of converting waste to energy on the planet. Here, let's take a close look at the Geobacter species. From the scanning electron microscope image, we can see it is a rod-shaped microbe flanked with narrow wires and long pili at the outer membrane. Geobacter species and syntrophic partners appear to be an important process in anaerobic wastewater digesters. Geobacter communities have proven to be a powerful approach to diagnosing the in situ physiological status of Geobacter species during groundwater bioremediation. Geobacter is known as a type of bacteria that is able to conduct levels of electricity, also known as electricogens, due to their ability to create an electric current and produce electricity. Geobacter species are generally the predominant Fe3-reducing microorganism in many soil and sediments, in which Fe3 reduction is an important process. According to literature, different Geobacter species reveal that they varied in their capacity for current production. The graph shows the current density comparison between Geobacter species. We can see that G metallic reducens and G sulfur reducens generate high current density. Thus, we are initially identified them as prototypes for future engineering.
Here is our conceptual model of waste energy on Mars. We hypothesize to turn organic waste on Mars into electricity. If the mission of sending humans to Mars is successfully achieved, organic waste will inevitably generate accompanied by human activities. Utilizing geobacter in the organic waste treatment seems a promising technology, and it can simultaneously realize the electricity generation by reducing ferric oxide in Mars regolith. Here is our geobacter full-cell reactor design. The anode is filled with geobacters, and the cathode is filled with the purified E-pili nanowires, also from geobacters. The aqueous organic waste on Mars will pour into the anode chamber for geobacter digestion. During this process, the anode will generate electrons, which can be transferred to the cathode. The cathode chamber is dispersed with Mars regolith, which contains 80% ferric oxide. The effluent of the anode and cathode chamber are carbon dioxide and ferrous ion solution respectively. In our future plan, a metabolically versatile bacterium, Pseudomonas palustris TIE1, can be further introduced to oxidize ferrous ion to ferric ion by taking carbon dioxide from the anode chamber or atmosphere and solar energy. Then the ferric ions can back to ferric oxide in a chemical way to achieve inner cycling electricity generation. We think there are 4 advantages of our geobacter full-cell reactor design. First, we can achieve the recycling of organic waste on Mars. Second, the full-cell electric generator can be a good complementary electricity source with solar energy, especially when sandstorms happen, which made the solar panel unavailable to work. Third, the ferric ion source can be directly get from Mars regolith, and is an anaerobic reaction. Oxygen can be saved for other space missions. Fourth, we also conceived a fully self-sustainable full-cell reactor with minimum source cost by introducing the Pseudomonas palustris TIE1 to oxidize ferrous ion back to ferric ion by intaking the carbon dioxide generated by geobacter waste consumption. In order to improve the electricity production, we decided to biologically engineer the geobacter string to achieve high current generation in the fuel cell. According to the pathway provided by KEGG, we found that the geobacter consumed organic waste and turned them into acetyl-CoA to start the TCA cycle, and then release carbon dioxide and free electron to the outer membrane. The electron can move along the pili through a metal-like conduction, and that can be attributed to the overlapping pi-pi orbital of the aromatic amino acids in the carboxyl terminus of pili. The methyl-heme C-type cytochrome OMCZ and OMCS associated with the pili, and they are essential for the ferric oxide reduction, presumably facilitating the electron transfer from the pili to the ferric oxide. Based on recent study, OMCZ nanowires were mostly responsible for the construction of the major portion of the electron transport pathway at the surface of the anode. Since OMCZ is a key component in electron transfer through differentiated geobacter biofilms to electrodes and gives the highest current densities. In order to increase the current density production, for the first approach we proposed is to overexpress OMCZ and pili gene to achieve that goal. Strong promoters may introduce before each gene and to ensure the highest expression. And we'd also like to generate a promoter mutant library to check the expression level by measuring the current density and ferric ions production rate under the ferric oxide environment. The second way we propose is to generate aromatic-rich pili geobacter species. The aromatic amino acids are considered to be important for electron transport. By searching the PLS genes of geobacter sulforidins and geobacter metallidins, we can find out that there are only a few aromatic amino acids, phenylalanine and tyrosine, which are shown in red on the gene sequences. In order to make an aromatic-rich E-pili, we proposed we first replace the non-structural essential amino acids with aromatic amino acids or synthetically generate polypili recombinant gene and introduce heterologous aromatic rich genes as linkers. We expect to increase the P-line conductivity by 5,000-fold to maximize electron transfer ability of pi-pi interactions. The third way we propose is to use direct evolution to generate high current protection strings. The concept was extended to protein evolution, while the evolution of bacteria under selection pressures that favors the evolution of a single gene in its genome. The Geobacter species naturally utilize selective pressure to optimize the reduction of other extracellular electron acceptors, which can be accelerated in the lab by using direct evolution, which may have fortunately led to the superior ability of Geobacter species to produce high current densities. The air from PCR can help in generating mutants in the species. The ferric oxide-rich environment can serve as the selective pressure to improve the current protection capabilities of Geobacter species.
The average temperature on Mars is minus 89.2 degrees Celsius, with a large range between minus 158 to 20 degrees Celsius. Almost all microorganisms cannot survive in the harsh environment on Mars. The low temperature and low pressure on Mars make water remain in the solid phase, while bacteria needs liquid water to do biological activities. Water is required to dissolve enzymes and soluble metabolites. Also, from kinetic theory, the chemical reaction rate is dependent on temperature. The low temperature causes slow growth for most terrestrial bacteria, and slow growth means slow reaction rate in the bioreactor. So the heat retention of the bioreactor is critical. In space mission, generally speaking, people demand retain heat for microbial growth. When bacteria grow and degrade organic compounds, they release some heat. So we want to retain this heat in the reactor. Therefore, during the startup and routine maintenance, less energy is required to heat the reactor. In space missions, although energy is generated through photovoltaic cells, it is a good practice to develop self-sustaining systems that require minimum maintenance. To retain heat inside the bioreactor, we propose to carefully design the reactor insulation. The heat generated by bacteria growth is considerable, 170 watts per kilogram of bacterial biomass. As shown in the example insulation experiment on the right, when the ambient temperature is about 5 degrees Celsius, a good insulation can keep the reactor at about 20 degrees. This is only using insulation materials as a cover on the outer surface. On Earth, the atmosphere is much denser than that on Mars. The Mars atmosphere pressure is less than 1% of that on Earth. So this thin atmosphere will help the heat retention. Due to the high atmospheric pressure on Earth, it is sometimes not feasible to use vacuum to insulate the whole reactor, but on Mars it may be possible. The startup and routine maintenance of this bioreactor are also energy intensive on Earth, but we want to minimize energy using during a space mission. During startup, it is possible to use a plutonium isotopic decay heating device to heat up the media and start the reaction. Plutonium heater was widely used in space missions as it is foolproof and reliable. It has been reported that Geobacter is resistant to radiation as it can clean up radioactive uranium waste. As 1 kilogram of bacteria can generate 170 watt of heat, if the insulation is good, minimal maintenance heat is required. We did some economic analysis on the launching cost. According to SpaceX, in the near future, it will take about $500 to launch 1 kilogram of payload onto Mars. The estimated weight of launching is as follows. A 1,000-liter bioreactor is about 1.2 tons. Insulation material plus electrode is about 100 kilograms. Piping and pumping will be 300 kilograms. The cost of launching is in total about $800,000. However, it is difficult to estimate the Mars environmental benefit in monetary value. Our project is based on the demand for a small environmental footprint during settlement constructions. In summary, we have generated a crazy idea of converting waste to energy on Mars by noting the composition of Martian relicus and the property of superbacteria on Earth, which is geobacter. We are attracted to the geobacter because of these highlights. First, geobacter grows under anaerobic conditions and can use iron as the terminal electron acceptor. There is no need for oxygen for this reaction. Second, the special pili can attract to insoluble ferric oxide, thus a minimum water content is required in the growth media. Since the water and oxygen are very precious on Mars, low demand for these two resources is a critical criterion for the large-scale bioreaction on Mars. Since Mars is a planet with very similar solar time to the Earth, there is no sunlight on Mars at night. If we build solar panels to generate electricity during the daytime, the biofuel cell could be a good complementary for electricity generation at night. The temperature is important for cell growth. However, the temperature on Mars is relatively low compared to Earth. Thus, we decided to design a self-sustainable temperature-controlled reactor to keep the steady operation of electric generation. Finally, thanks for watching, and we welcome your comments in the comments section below.